Hello, I am Sage. Welcome to today's Journal Club. We'll be critically examining a recent preprint from the Zoo Lab at Stanford, introducing the virtual Biotech, a multi-agent AI system designed to mirror the organizational structure of a biotech company for computational drug discovery. This is a preprint published online in February 2026. Here's our roadmap. We'll start with the problem the authors are trying to solve, walk through the system architecture, then examine each of the three applications in detail. I've reserved dedicated slides for methodological critique and limitations. The authors frame their work against a well-documented problem. Drug discovery has staggeringly high failure rates. About 90% of candidates entering phase 1 never reach approval, with efficacy and safety accounting for roughly 79% of failures. The key bottleneck they identify is fragmentation, different teams working with different data types in silos, missing cross-referencing opportunities. I want to ask the group, do you think fragmentation is really the primary bottleneck, or is it more fundamental biology we don't yet understand? Here is Figure 1, the core architecture. Panel A shows the organizational chart. Note how it explicitly mirrors a biotech company with divisions for target ID, safety, modality selection, and clinical operations. Panel B shows the MCP tool servers grouped by domain. Panel C illustrates the workflow. User query goes to the CSO, who consults a chief of staff for briefing, clarifies user intent, then routes to specialist agents. The scientific reviewer then evaluates outputs before the CSO synthesizes. A key question for discussion: does this organizational metaphor genuinely improve performance, or is it primarily a framing device? The paper doesn't compare against a single agent baseline. The scale of data integration is genuinely impressive. Over 100 million single-cell profiles, billions of perturbation measurements, and tens of thousands of clinical trials, all accessible through standardized MCP interfaces. The MCP approach is notable because it abstracts away heterogeneous APIs into typed, documented tools that agents can discover and compose. However, a good question for the group: how much of this data does the system actually use in any given analysis? Having access to 14.5 million protein interactions doesn't mean the agent uses them effectively. The orchestration design is one of the paper's most interesting architectural choices. The CSO never touches data directly. It only reasons about what to ask, who to ask, and how to synthesize. The scientific reviewer creates a feedback loop that can trigger reanalysis. The supplementary notes show a detailed example of reviewer feedback for the OSMR case. It's remarkably thorough, flagging 6 specific issues including missing benchmarks and unjustified ratings. A good discussion point: is this reviewer agent genuinely catching errors, or does it just produce the appearance of quality control? Figure 2 shows the extraction pipeline and resulting dataset. Panel A illustrates how each agent follows a 3-tier evidence cascade: first clinicaltrials.gov, then PubMed, then web sources. Panel B is the Sankey plot, showing the flow of 55,984 trials across phases and outcomes. The 184-fold speedup from parallelization is striking. Note the practical design choice. Phase 1 success was defined algorithmically as progression to Phase 2, since Phase 1 trials assess safety and dosing rather than efficacy. This is a reasonable simplification, but worth noting, it conflates regulatory and sponsor decisions with scientific merit. The validation is reasonable, but has caveats worth discussing. Only 100 trials were manually reviewed out of 55,984. That's 0.18%. The 89.7% primary endpoint agreement sounds good, but note it's 78 out of 87 applicable trials, meaning some trials were excluded from the denominator. The authors argue disagreements reflect genuine ambiguity in binary classification, which is fair, but ask the group if the agent disagreed with the human reviewer in 10% of cases, and those cases are the most clinically nuanced ones. Does that matter more for downstream statistical analyses? Figure 3 is the centerpiece of Application 1. Panel A shows the forest plot for tau cell type specificity and bimodality, coefficient against multiple trial outcomes, phase progression, termination, and endpoint success. The effect sizes are modest but consistent across outcomes. Panel B shows the adverse event analysis. Higher specificity correlates with lower AE rates across organ systems, which is biologically intuitive. More specific targets should have fewer off-target effects. This analysis links in vitro perturbation responses from Tahoe 100M to clinical trial outcomes in oncology. The 6 hallmark scores, apoptosis, proliferation suppression, DNA damage, stress, resistance, and cell cycle arrest, were computed from log-fold changes of curated gene sets. The DNA damage score showing the strongest association with phase 1 to 2 progression is interesting. The authors suggest this may reflect the importance of demonstrating pharmacodynamic activity in early phase studies. A critical note, the gene sets are quite small, only 5 genes for DNA damage. How robust are these composite scores to the specific gene selection? The functional genomics agent chose these genes based on canonical roles, but alternative gene sets could yield different results. Figure 4 is the most visually rich figure in the paper and tells a compelling story. Panel A shows the agent's reasoning about absent germline signal. Critically, the CSO recognized that for checkpoint targets, therapeutic rationale comes from somatic tumor overexpression, not germline risk variance. This is genuinely good reasoning. Panels B and C reveal the surprising finding that B7H3 is most strongly upregulated in cancer-associated fibroblasts rather than malignant cells. This is a less explored angle than the cancer cell-centric view in existing literature. Panel D shows ligand-receptor interactions, Panel E the spatial immune exclusion, and Panel F survival analysis. Question to the group: how much of this discovery reflects genuine novel reasoning versus the agent accessing datasets that would lead any analyst to the same conclusion? This flowchart traces the evidence chain that led to the ADC recommendation. What's notable is the reviewer-triggered handoff at the bottom. The scientific reviewer flagged that ligand receptor inferences from dissociated single-cell data lacked spatial context, which prompted the CSO to dispatch spatial transcriptomics analysis. This is the kind of cross-division integration the authors argue siloed human teams would miss. The survival analysis shows B7H3 as a prognostic biomarker with HR 1.82, though the confidence interval is wide, 1.06, 3.15, and the p-value is 0.031, not highly significant. A discussion point. The authors conclude B7H3 acts as an independent prognostic biomarker, but this is from a single cohort analysis. How reproducible is this finding? The authors present the FDA Breakthrough Therapy designation for ifinatamab deruxtecan as external validation. This is compelling framing, but let's be critical. B7H3 ADCs were already in clinical development before the systems analysis, so converging on ADC as a modality isn't surprising. The real novel contribution is the CAF-centric mechanistic hypothesis, which the FDA designation doesn't specifically validate. Also, while the authors disabled web search to prevent information leakage, the underlying LLM was trained on literature through January 2025 that discusses B7H3 as an immunotherapy target. The question for the group: does the system's reasoning genuinely add value beyond what was already known in the literature it was trained on? Figure 5 shows the full OSMRV case study. Panel A shows the agent's reasoning through the trial protocol. Panel B is particularly interesting. It shows consistently higher baseline OSMR expression in non-responders across 5 independent UC biologic trials, testing 3 different therapeutic mechanisms. Panels C and D dive into single-cell analysis of the Taurus dataset. Panel E proposes the biomarker-guided enrollment strategy. This case study is arguably the most clinically actionable application because it produces a concrete recommendation: screen patients for OSMR expression before enrollment. However, the agent's analysis is entirely retrospective, and we don't know if biomarker-guided enrollment would actually have changed the outcome. The cross-trial OSMR finding is one of the most compelling results in the paper. Across 5 independent trials with 3 different mechanisms of action, anti-TNF, anti-IL-12/23, and anti-integrin, non-responders consistently showed higher baseline OSMR. The Taurus single-cell analysis then pins this to specific fibroblast states that show increased JAK-STAT activity and 3 coordinated transcriptional programs. However, note that the Taurus finding about OSMR in non-responder fibroblasts came from the original publication's supplementary materials. The agent found and synthesized existing data rather than generating entirely new analysis. Is that still valuable? I'd argue yes. Connecting this existing single-cell finding to the cross-trial biomarker evidence and the Moonglow trial design gap is novel. On the strength side, the authors have been thorough. Permutation testing, mixed effects models, adjustment for genetic evidence, and a thoughtful knowledge cutoff design for the B7H3 case. The replication of prior genetic evidence results lends credibility. On the concern side, several stand out. The observational design means we can't establish causation. Cell type specificity correlates with trial success, but is it causal or confounded by target class? The k-means binarization is arbitrary, and no sensitivity analysis is reported. Only 100 of nearly 56,000 trials were manually validated, and for multi-target drugs, using the minimum tau across targets is a strong assumption. Asking the group, which of these concerns is most problematic for the paper's conclusions? These are the limitations I think are most important to discuss. First, missing controls. The paper never compares the multi-agent system against a single agent baseline or a human expert team performing the same task. We don't know if the organizational structure actually helps. Second, AI-specific risks. Hallucination can propagate through the pipeline, and despite the reviewer agent, there's no quantification of error rates in the reasoning chains. Third, generalizability. 3 case studies is a small sample, all chosen retrospectively with favorable knowledge cutoffs. Publication bias is a real concern. We only see the successes. Finally, a philosophical question. Can this system discover something genuinely novel, or does it recombine existing knowledge in useful ways? Both are valuable, but the paper's framing sometimes implies the former. Let me give my verdict for the group. The central claim that the virtual biotech enables more transparent, efficient, and comprehensive analyses is partially supported. Cross-modal integration and transparent audit trails are clearly demonstrated. The single-cell feature findings are genuinely novel and well-validated statistically. However, the efficiency claims lack rigorous comparison. The case studies are retrospective with favorable selection, and the paper never demonstrates superiority over simpler alternatives. 3 questions for discussion. First, is the organizational metaphor actually improving performance, or is it just a framing device? Second, should pharma companies adopt this approach now based on retrospective demonstrations, or should they wait for prospective validation? Third, what regulatory framework would govern AI-generated therapeutic hypotheses? These are the questions that will determine whether this becomes a transformative tool or an interesting proof of concept. Here are all the key references cited throughout the presentation. The primary source is the Zhang et al. Preprint from February 2026. I've also listed the major validation datasets and clinical trial references that support the three applications discussed. To wrap up, this is an ambitious paper that showcases what multi-agent AI systems can do in drug discovery. The cross-modal integration, particularly the B7H3 KF-centric hypothesis and the OSMR cross-trial biomarker finding, represent genuine analytical value. But the field needs prospective validation and head-to-head comparisons before these systems can be trusted for real drug development decisions. I'd love to hear the group's thoughts, particularly on whether the organizational metaphor adds real value and how you'd design a prospective test of this system.